Starting. It says we are live. And so I'm just going to wait for my little YouTube page and for the peoples. Yeah, I think we are started. We are here. We are ready to start this show. Without any further ado, in 3, 2, this is TWIS. This Week in Science, episode number 713, recorded on Wednesday, March 20th, 2019, Fishing for Science. Hey everyone, I'm Dr. Kiki, and tonight on the show we are going to fill your heads with magnetism, old sperm, and bait. But first,
disclaimer. It's all in your head. What am I talking about? Everything. Everything that is or isn't real. Everything that is or isn't possible. Everything that has ever happened or will ever happen is all in your head. Yes, there is a world on the other side of the skull wall that goes on regardless of what's going on inside. But as far as you go, nothing comes closer to the reality of your existence than what takes place between your ears. And if you happen to be a scientist or wish to think like one, the most intuitive instinct you must overcome is trusting what is in your head. The most important training you will ever undertake is learning to trust what is on the other side. While this is traditionally something that takes oodles of hours of patient practice to master, we have devised a method to accelerate the process. An injection of scientifically formulated information applied directly to your ear holes that we call This Week in Science. Coming up next.
I've got the kind of mind that can't get enough. I wanna learn everything. I wanna fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I wanna know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
and a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are back again to talk about all sorts of science news that has happened over the last week, and there's been a lot, as usual, but we whittled it down. We have whittled and wheedled and figured out what we wanted to talk about this week, and so we hope you enjoy the show. What did I bring? I brought stories about brains, mini brains, and we have an interview about brains.
That's good.
Kind of my wheelhouse. I kind of like it. Makes me feel comfortable and happy and warm inside. It's nice. It's good. Jello-y brains. Justin, what'd you bring?
I've got the answer to where gods come from.
Oh,
Finally been answered.
Oh,
And
great.
how hepatitis B can do science.
okay. I am really interested in both these stories. Can't wait for that. Blair, what is in the animal corner?
Well, I too decided to stick to my wheelhouse tonight. I'll be talking about old sperm and bird poop.
There we go. Your wheelhouse is fun.
Yeah,
I'm staying away from your wheelhouse.
it's sticky sometimes, but it's fun.
Off to a rolling start. Everyone, as we jump into the show, I want to remind you that if you have not yet subscribed to This Week in Science, you can do so to find whole episodes that get downloaded to your device, whatever it might be, at twist.org. You can find us on any podcast player, whether it's Google, Apple, Spreaker, Spotify, or Pandora. All of them are out there. You can also find us on Facebook and YouTube. And if you're in Portland, Oregon on April 3rd, you can meet us live in person. We are doing a live theater podcast recording It's going to be fun. We are going to be joined by musical guests, the PDX Broadsides, and local to the Portland metro area, Washington State University, Vancouver scientist Allison Coffin, who studies hearing. Going to be a lot of fun. Get your tickets. You can find out at TWIS.org. More information. All right. Ready to move on? Yeah. Okay. Let's jump into the interview. Our interview tonight is with Dr. Chris Donnelly. Dr. Donnelly is an assistant professor in the Department of Neurobiology at the University of Pittsburgh School of Medicine, and he's the scientific director of LiveLikeLou Center for ALS Research in the University of Pittsburgh Brain Institute. His studies are focused on the biology underlying neurodegenerative diseases with a particular interest in understanding neuronal death observed in amyotrophic lateral sclerosis, ALS, and frontotemporal dementia, FTD. Chris, welcome to the show.
Hey, how you doing?
Wonderful. How are you?
I'm good. How are you? How's everyone doing?
I would love to know. So many of us have heard about ALS either because of people that we know or, social media is huge these days. And a couple of years ago, the icebuck challenge was a massive social media effort to raise money for ALS research. But can you give us some insight into how you got interested in studying this particular disease?
Yeah, so I got interested in ALS when I was like 15 years old. In fact, we had been assigned to read a book called Tuesdays with Maury in high school, and it was just something we had to do for an English class. And at the time, I didn't know what ALS was. I don't think it was very well known at the time either. And I just read about this person who went to visit a mentor of his. It was sort of a biographical story, and chronicled how this person's mentor sort of went through the disease progression and eventually passed away from it. And as like a 15-year-old kid who, didn't understand mortality, I don't think at the time, it was very frightening. And, as I've kind of continued my research, my scientific career, or just like education, it was always something that I was really interested in, but didn't really get into it until after I got my PhD. That's really
Yeah. And so your work has gone from like stem cell focused to being specifically like protein focused. Can you give us a little overview of the path that you have taken to get where you are?
Yeah. So you're speaking to someone who was like one of those people who had multiple majors in colleges and never could figure out what they wanted to do, but always liked biology, sort of ended up sticking with it. Had a really great mentor who was in undergrad who was teaching me about DNA and like DNA epigenetics, and how DNA opens and closes, and you can open and read certain genes. And she got me interested in molecular biology, suggested I go to graduate school where I studied like nerve regeneration after an injury, but really looking at like molecular side of things. And then at that point, after I was kind of finishing up, I had the opportunity to finally get into ALS, and then went and worked and trained under some ALS experts at Johns Hopkins. And that sort of was my personal history. But while I was there, that's when I sort of learned like all about stem cells and how we could use these as models for the disease and whatnot. But yeah.
How can you use them as a model for the disease? I mean, how do you take just a cell and go, this is what we learn from?
Yeah. So, these neurodegenerative diseases, the major problem is, we don't know what causes them. So how do you study them in the first place? If you don't know the cause, you can't, it's not like a tumor. You can't really do a biopsy like you can with some cancers. So, when these induced pluripotent stem cells is a technology that was developed in, sort of the early mid 2000s. And this technology basically was able to reprogram these skin cells or blood cells and kind of trick them into becoming embryonic-like. And then you could kind of trick them into going into development. And for the first time in human history, you can look at human neurons, like a live human neuron. And so this opened the field really for neurodegenerative disease research, because now you can actually look at neurons and study them from a person who has a disease, especially for a disease you don't know the cause of. And for ALS, for example, most people who have the disease, 90% of them, of patients, there is no, family history or genetic cause. Most people who have it, that we don't know why they have it. There isn't a genetic reason for it.
Oh, that's fascinating. So it's not in most things I just consider it's going to have a massively genetic component, and so this doesn't.
Yeah, I mean, there's risk factors that we have,
Yeah.
but for the most part, 85 to 90% of patients who have ALS have no family history of the disease. And it's even less for things like Alzheimer's, for example. So it's very frustrating as a scientist to how do you study something that you have no way to model it. So what people have done historically is they've like taken a model, could be anything from a yeast to a mouse to a neuron from another animal, and you put in a mutation that we know can cause the disease, and then you just make a lot of it, and you try to see what that's doing to the cell. The problem is that that's not what really happens in most patients, and so there's kind of been this lack of translatability, I think, between like the model and the actual disease itself. So yeah.
So, like yeast, I would think would be a pretty decent model, just in that on the genetic level, which I guess is not being a genetic disease, but that the propagation of new neurons and the budding of yeast use a conserved set of genes to do a lot of these activities.
Yeah, absolutely. Yeah, you're 100% right. And that's the really cool thing you can do with yeast and a lot of these smaller organisms like flies and things too, is that we have the genetics down so tightly. We know so much about them. We can manipulate things so quickly and easily. Within weeks, you could have knocked out almost every gene in their genome and see what is correcting that genetic mutation that you put in. So if you put in a mutation that causes an ALS mutation that causes a yeast to die, and now you've just selectively started knocking out different genes in those yeast, and you hit every one of the genome, and you can now find hits that actually protect against that death.
Wow.
Right. Yeah. And you can do the same thing with, flies and other rodents. And you're now with CRISPR, which I'm sure you guys are very, very familiar with now. You can do it even in like human cells and rodents too now.
Yeah. So, I mean, you talk about yeast and flies, and it's the kind of limitation is the life cycle. So how fast does that cell division and the developmental cycle take place to let you actually know what's going on?
Yeah.
And so with human cells, is there a like what's the time scale compared to, I mean, human cell studying, obviously it's best because that's the system that we want to understand. But is there a time scale difference between yeast and flies and those other things?
No, you're so right. So you can kind of extrapolate how old a mouse or a fly is compared to a human based off the life cycle. But, with diseases like ALS, the average onset is 55 years old. When we make stem cells, it takes a couple months, excuse me, maybe 3 months or so to make neurons, human neurons from stem cells. And so even then, though, what you're looking at are really, 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 really young neurons. Like
they're not 55-year-old neurons.
No.
So you have to wait 55 years before conducting your study?
They haven't had a job, been fired. No.
You're dealing with like third trimester neurons. They're crazy. So, it's kind of interesting. Like, how do you age a neuron in a plate to get it to be 55 years? It's sort of a conundrum. But yeah, and it's actually interesting if you ever look at them, compare them to, say, a mouse neuron, like an adult mouse neuron, and you compare an adult mouse and a stem cell in the same dish, the mouse neurons are usually larger because you're looking at such young human, neurons. It's kind of interesting, but.
And so you're looking at things like we're talking about just the basic stem cell kind of genetic editing aspect. CRISPR, you can knock things in, or just with other technologies also, you can do knock-ins and knockouts and figure out what is part of the genetic system. But you've taken things a different direction and are not looking necessarily at the genes, but at the proteins. And can you tell why you started doing that?
Yeah. Oh, thanks. So thanks for setting me up there. That's great.
You're welcome.
So Yeah, so the one thing that you can say about almost every neurodegenerative disease is that regardless of if they have a genetic cause, for the most part, there's always an exception, that almost every patient, when you look at the brain or the spinal cord after they had passed away, looks like they have the same, what we call neuropathology. And what that means is that there are features of the brain that you don't really see often or as severe in healthy people who've died of just old age. So in ALS, for example, and most of these diseases, this manifests as like these proteins that clump. And depending on the disease, the protein differs. So for example, Parkinson's disease, you have something called synuclein that clumps in the neurons that are dying, in the region that is dying. In most dementias, you have something called tau. And in Alzheimer's, you have tau and something called A beta. Right. And in ALS, you have a protein called TDP43. So no matter what the cause of ALS is, almost every patient, with the exception of maybe 3%, and they usually have a genetic mutation, have these protein clumps of this protein called TDP. And it should be in the nucleus of the cell, doing what it does a normal job in processing RNAs and working with the DNA. But it's actually outside of the nucleus. It's missing from the nucleus. And in most cases, it's actually forming these clumps. And so the problem is there's really no reliable way to recreate that. And so what we've been trying to do is actually develop a method to sort of turn on that pathology, that thing that almost every ALS patient has, and try to kind of reverse engineer what that's doing to the cell and why it's happening.
So it's not necessarily an overproduction of, but it's actually expressing, like somehow it's getting moved, like there's there's something transporting it.
Yeah,
which shouldn't be. So it could be like a transporter that's like,
I love this. You're setting me up again. No, and actually that's another big area of study in general in the field. Is normally this protein can kind of cycle in and out of the nucleus, but one area of interest is, maybe it's getting stuck on the outside of the nucleus, and that's why it's getting these clumps to form, or why these clumps are forming.
But okay, so we've talked on the show before about, and I always get the pronunciation wrong because I study birds also, and so prions.
Yeah.
The misfolded proteins that cause scrapie and mad cow, and then there's also like you mentioned, these misfolded proteins that are involved in Parkinson's and Alzheimer's. What is going on with, like, is there a common thread that's being found through all these diseases for this misfolding?
Yeah, so it's really interesting. A lot of these proteins that undergo this misfolding have prion-like domains, and they're just sort of a part of the protein that likes to get stuck together. It likes to clump, and there's some biological reasons for that. And one of those is that these proteins, when they're around proteins that they like in the cell, they can kind of what we call phase separate out of the cytoplasm or out of the nucleoplasm. So they can almost like form mini organelles without a membrane by being attracted to one another. The problem is if that goes awry, then these things can start to get stuck together.
Right.
And that's where the question is, why does that happen? Why do we go from something that's normal and healthy to this sort of, solid structure that's kind of aggregated is what we call it.
And then it gums up the works and gets in the way of normal cellular metabolism.
Yeah, and we don't know, for example, like a lot of these proteins, we don't know if they spread. Depends on the protein. Like we don't know if they spread like some of these prion proteins, but we think that they might, or at least they have the capacity, because it does seem most of these diseases seem to have like a spreading pattern when you look in patient tissue and whatnot. So
I think, that's something that I've always found kind of interesting, is the idea is that that in some of these disorders, that once it gets started, it's a cascading effect because the proteins actually in their interaction with each other cause more misfolding.
Yeah.
that there's some kind of dynamics going on, that it's like, you want to be like this.
Yeah. And the craziest thing, we work a lot in our lab with human tissue. We try to recreate these in the lab using light, which I could mention if you want. But the idea is that, we're trying to recreate these problems. But what's interesting about it, and I don't say interesting, but what's kind of scary about it in retrospect is that, when we take and work with these proteins, like, are they contagious like prions? Should we be taking special cautions that we didn't really think about because they're not really in existence now? We don't know if they spread like those. So it's kind of interesting just from
because what you're describing isn't like a rogue ribosomal thing where they're getting printed in this misfolding. You're describing that this is post-healthy proteins then interacting with other proteins that are somehow convincing each other to fold differently.
Yeah, so I'm getting a little jacked up, so you have to stop me if I'm getting
get in it. That's what we like.
So one of the things that we were trying to do with we just published a paper recently, and one of the things we were trying to do is to recreate these clumps that we see in ALS specifically. And so what we did was, because there's not really a good way to do this in a live cell, we genetically engineered the protein so that it had a tag on it that responds to light. And what would happen is, if we put this in, made this express in the cell, human cells, shine light on the cells, the tag comes together. It's from a mustard plant, and it normally is a phototrophic protein. So this domain will help the plant move in the direction of sunlight. But we don't really have this domain in mammalian cells. So we put that domain on this protein TDP, we shine light on our cells, and we were actually able to sort of recreate these clumps that we saw in the patient tissue. And we did all this validation, and they actually kind of propagate a little bit, at least within the cell. It's kind of interesting. They recruit more of its own protein. It starts to pull in the endogenous, and they're neurotoxic. They kill the neurons, which was really interesting. So it was kind of neat, because now we have a way to actually turn on the disease with light, or this key feature of the disease, by just shining light on these cells.
Wow. So now you have a model system where you can turn the disease on. Can you turn it back off again?
Loving this. Yeah. So, so what was really cool is that it puts us in a position to almost reverse engineer it. Why does this happen? If we shine light on these cells, why is this getting stuck together? And what we found is that when we just shine light on these cells, the protein is in the nucleus, but it starts forming these clumps in the cytoplasm. But the light was all over the cell, so it didn't make sense. But what's interesting is that the nucleus has a lot of RNAs and nucleic acids, as you guys know. But the cytoplasm, it has RNAs and nucleic acids, but they're usually already interacting with things. And this protein, its main job is to process and interact with RNA or DNA. And what we found was that the protein that aggregated was the form of the protein that wasn't interacting with its normal partner. So the problem wasn't that the protein itself was getting stuck to itself. It just couldn't get stuck to itself if it was doing its normal job. And that was really interesting, because it kind of leads this idea that there are these natural inhibitors in the cell that prevent these things from clumping. And we just kind of have to reverse engineer it, or think, well, what are we missing? If we're seeing things clump, that means that something in the cell is not interacting with it to prevent it from clumping.
Right. It should be interacting with something else. It has a different job to do. But for some reason, it's not making that connection.
So now it's hanging out and on the corner with the rest of the delinquents, and they're forming gangs of ne'er-do-wells because there's not enough jobs.
Exactly.
So now you've got to find the missing RNA or whatever it is that they're normally interacting with. That's the deficit that's causing. It's not even the protein's fault. This is just the environment that it's in. It's doing its normal job.
Yeah, totally. And the cool part too about this, I think, is that one of the things that we were able to do is we said, well, if we know that the RNA is the problem, could we make something that looks like what this protein likes to interact with? So could we make a fake version of it? We call them baits because it's like fishing for the one that's the target. And so we just made some, we put them in the cells, and now we actually were able to prevent these things from clumping in the first place, even if we tried to get it to happen. But and so that was really exciting because like the type of molecule we're using is already a therapeutic, it's a therapeutic molecule that's being used now. And so we just kind of made our own version of it, and it seems to work. So for all these degenerative diseases, we're kind of hedging our bets on that. There are natural inhibitors in the cell that prevent these things from clumping. If we can find what they are, we can just kind of create a bait or some sort of molecule to fake its normal partner and prevent them from actually aggregating in the first place.
And this kind of gets at the idea that, like you said, there are some 90% of ALS patients are not genetically predisposed, that maybe they're environmentally or, there's other factors involved in the development of the disorder. And so maybe this is getting at it. There's something that's going on in the environment or whatever they're doing that is changing certain and microRNAs and these little messengers that go in and connect to these things to allow upregulation, downregulation, silencing, all these little factors. Those are very environmentally controlled.
Because it would have to be, because what we're describing is something that it would make total sense that this could one-off in a cell here or in a cell there. But for it to be this dominant across cells means that there has to be an environmental factor that's dictating to all of the cells to inhibit this RNA creation, right?
Yeah, and the thing is, there's probably some hints that we can take from just what we know from patients. We know that military veterans, whether or not they served during a time of peace or war, U.S. military veterans, are 2 to 3 times more likely to develop the disease.
Wow.
Athletes, people who are really highly physically active, have an increased chance of getting the disease as well. We do know there's some increased prevalence in NFL. We know there's some increased prevalence in certain soccer players in Europe, but certain countries, it just depends. So maybe there's something about activity that's creating this. Maybe, activity or lack thereof is creating RNAs that are, or not creating RNAs that normally sort of bind this thing.
One thing, all those groups
stop working out. I'm getting loud and clear. Got it.
That's not what he said, Blair.
Early and work out, whereas everybody else gets to sleep late. Maybe this is all.
Yeah.
I'm interested also in the link between ALS and frontotemporal dementia. I mean, and then from there, even the Alzheimer's. These are, like you said, there are multiple different proteins involved in different disorders. But are ALS and frontotemporal, are those very closely linked?
Very closely. So there are there are certain families who have genetic forms of ALS, for example, or genetic forms of FTD and frontotemporal dementia, and you can trace that there are some family members who will get one, some family members who will get the other, and some who will get both. And in fact, there's I think it's like 30%, 20 to 30% of ALS patients will develop a dementia, frontotemporal dementia, later on in the disease, and vice versa with FTD.
Like insult to injury.
So
I know.
Can we slow down for a second? Can we explain what that is?
Frontotemporal dementia?
Yeah.
So it's a dementia, but it's not an Alzheimer's dementia. So what it is it's dementia of the frontal and temporal lobes. And what that means is it's not so much a memory loss, but it's more like personality change, sometimes speaking difficulties, social kind of distance, loss of empathy. So things like, kind of think of the mean old grandfather all of a sudden, that type of thing. And it's I think it's the second most common type of dementia in the US. Sometimes it's misdiagnosed for Alzheimer's, but it usually hits under 65. Yeah.
So it hits similar to, like you said, ALS hitting around 55. It's a younger disease.
Yeah.
Something that's more older.
Yeah, and then there's certain a couple years back, there was a really big discovery where we knew that there was this clinical overlap between these diseases, and we found some really well-known people at different institutions found that there was a mutation that could cause either ALS or FTD. And it just happens to be the most common genetic cause of both of these diseases now. And you can find it in about 1 in 8 patients with ALS. Similar, I forget the exact number with FTD, but it's similar. And there's families, in fact, there was a group of twins, a set of twins at NIH who both had this mutation, identical twins, I believe. Could be wrong here, but they were definitely twins. And one developed ALS and one developed FTD.
So somewhere within this mutation then is perhaps, right, the focus of what the affect of that is downstream, which must also be environmentally able to be impacted for all the rest of the cases. But in a way, they found this is perhaps the genetic pathway that by itself is like a knockout or an added, however you want to look at, is something in this is the cascade.
Yeah, so actually the thing that you brought up, the nuclear transport, that was one of the things that was found to be a problem in this type of ALS mutation. But then to add just real insult to injury, with ALS, there's maybe 40 different genes that can cause it. Mutations in up to 40. And so, and then if you looked at, let's say we take the first gene that was discovered, which was in, I think 1993, and we found 150 different point mutations, and all you need is one. So when you're modeling this disease, it's like you've got different genes, and in each gene you can have multiple different mutations, one of multiple different mutations. And so it just makes it really complicated to study. And that's only 10% of the disease. And so the rest of patients do not have those mutations.
So do you have access to, or are you this is like, because RNA sequencing is now where we're getting to the point where we can start engaging in proper RNA sequencing in larger volumes, and it's a whole next level of, oh my gosh, that's more complexity. But is this something that you've been able to start working on, or?
Yeah, so there's a lot of groups doing RNA sequencing like in patient tissue with stem cells. We are getting into it, but it is very costly. It's getting less costly. So we need one of those things where you kind of need a reason to get into it. But yeah, absolutely. I mean, it's really revealing a lot. I think the big point here is that with all these disease, with ALS and with FTD, you know, they're just really complicated, and it's very hard to study them. And you kind of have to pick your best model. You have to pick what you want. I mean, the only other organisms that I know of that get ALS are dogs, horses, and pigs. I'm not sure if there's any other animals. I'm sure there are, but they're the only ones that I can kind of spout off the top of my head that develop ALS naturally. So beyond that, you really have to kind of take one of these mutations, put it into an animal or put it into a cell type or something, and then just make a lot of it and try to study it. Or you could do what we did, which is a little bit different, but that's a whole new beast.
Yeah, take the proteins and turn on the disease in an animal, and then see if you can turn it back off again.
I mean, exactly.
Is that something that, I mean, now that you've done these cellular studies, are you interested in moving forward to see, are there drug targets? Are there drugs in the market already that could be used for this purpose? Is there that direction to move, like, you know, like you're saying, this is aimed at the much larger body of people who come down with this disease. So are you aimed at moving it down the pipeline toward a treatment?
Yeah, absolutely. I mean, the one thing I will say is that I'm a very firm believer of collaboration and sort of knowing what you're really good at, knowing what other people are really good at. And I can say I'm absolutely not a drug screening person. I've never done it. I'm not good at it. So from our end, what I would like is to really just kind of have a lot of people use the system, potentially companies as well, whoever has just the ability to do this efficiently and properly. You know, I think with ALS, it's such a aggressive disease, and FTD, because about 50% of FTD patients also have these protein clumps of TDP. And you see it in some Alzheimer's too, and some other diseases. But I just think that because of the aggressiveness of it, and if we want to move it quickly, which, you know, that's what patients want, I think we need to have the right people using it. So yeah, we're trying to make it very simple to allow a lot of people to access to it. And then hopefully it just takes on, because we do think it's a pretty big deal.
It does sound like a big deal. I mean, and like you said, there are a lot of people who could benefit from this. And aside from, your own laboratory next steps, I mean, I'm curious. There's a lot of work looking at like Alzheimer's and other brain dementia, degeneration type diseases that come from maybe blood flow changes where there's maybe, the filtering system isn't working the way that it should. I mean, are you
brain lymph
seeing
nodes?
the lymph node? Yeah, the lymph system.
Which got
carrying the
discovered
waste-
like what now?
Yeah.
5 years ago? Was it? Like we have this brain garbage collector we didn't even know we
That's
had.
right.
Yeah.
Yes.
Yeah, that's a great question. I admittedly haven't seen a lot of that. I know there are people working on the blood-brain. There are some people doing blood-brain barrier work because there does seem to be a breakdown there. But I haven't seen much. Right now, the really hot thing is microglia. That seems to be the really big thing, these immune cells in the brain that are responsible for inflammation. And you see inflammation in almost every neurodegenerative disease. But the idea is that these could be like really good targets to stop. They might be protective. They might be bad. I'm not 100% sure about it, to be honest, but I think it's a really intriguing area of research. The interesting thing about,
No, but you've completely enlightened me on this. Is I had thought of these neurodegenerative protein-related diseases as being a protein production problem, and it doesn't sound like what you're saying. It doesn't sound like it's an overproduction. It sounds like it's normal production, and that these are normally present proteins, and that they're just having a different affect than they normally would. And so the whole idea that we could solve it through the garbage cleanup doesn't seem as applicable if they were supposed to be doing a function there in the first place and have been repurposed towards the disease.
Yeah, I have to be careful because I'll get myself in trouble if I say too much about that. But yeah, so I agree with you. Like the autophagy, the degradation system, I think is going to be very important because your cells can handle a certain amount of trash or these clumps that you get in the brain. There's no question that cells can handle a certain amount, and this degradation system is responsible for processing that. The problem is, though, that with a lot of these, I think there's two things. One, if there's one or two major things that are clumping, it doesn't necessarily explain why not everything is clumping. Like if that whole degradation machinery is not working, then you would imagine there would just be cellular trash everywhere. And maybe in some diseases that's the case, but in like ALS, for example, is really one major protein.
It's very specific. Yeah.
Yeah. So, I mean, to me, I think that it's good to have that pathway kind of working really, really well. But if you have these clumps starting, what we typically see is that once they start, they kind of keep growing. And so you need to figure out a way to stop them from like continuously sucking in more of itself. It's like a black hole of trash, protein trash.
Right. It's going to be that gyre, the gyre that collects all the plastic in the ocean. Cellular protein clumps all floating around in there. How did you decide to aim to start using optogenetics and these light activated proteins in your research?
The proper answer is that I had a really good conversation with a colleague, and we were sitting down. Yeah,
but the real answer, yes.
I think we were in a bar over a beer at some point, and they were like, "Hey, I'm working with this protein that comes together when you shine light on it." And I was like, "Oh, that would be really interesting if we could try to do this." And lo and behold, a year later or so, 2 years later, here's where we're at.
I've always thought. I mean, I understand the usefulness of optogenetics in a dish, when you've got cells in a dish and you can turn it on and there goes cellular, you could make proteins, all this stuff happens. Awesome. Or you turn something off. Ta-da, that's great. But what about an animal or a person? Like how do you go from the dish to turning it into a system where you're, I don't know, you put it in, like you mentioned, maybe pigs. You're looking at the ALS system in pigs. How do you do that?
So people use them quite often in a lot of different animals. Most of the work is used in activating or turning on or turning off channel rhodopsin, which is a channel that you can essentially open up or close based on shining light, and either activate or turn off specific neurons. So it's pretty regularly used in like systems neuroscience to try to identify what circuits are responsible for things like rewards, reward behavior, things like that. So addiction research. I have a couple colleagues doing that actually, so they'll kill me if I don't mention it.
Right.
But so I think it's pretty well established. I mean, what people actually do is they'll use either like viruses or they'll make transgenic animals. And depending on the animal, like one thing that we've done is we made a fly for this because flies are easy and they're fast. And so we just took a fly. It's a normal fly. We genetically edited it, and then we kind of make it live in blue light when it's an adult. And now it develops what looks like an ALS phenotype in a fly. But it's, it was as simple as just exposing it to blue light. With animals like mammals and things that aren't as small, a lot of times you kind of have to go in and put in like a light maybe on the skull, kind of do a little surgery. But it's done pretty frequently nowadays in neuroscience.
This is this optogenetics when it became a thing a couple of years back. It's probably more than a couple of years now. I've been doing this too long, but I was just like, what are we doing now? This is amazing.
Yeah, it is crazy, right? Using light to control like some aspect of a cell. It's fascinating.
Yeah, turn it on, turn it off. Click it on.
The trick is to find a wavelength that'll penetrate the skull.
Yes.
Yeah.
Then you'd really be up to something.
Yeah, I mean that, but then you're also up against, like a wavelength that doesn't stimulate the way that normal visible light stimulates, because you don't want it, you want it to be something different from normal visible light, which is why the rhodopsin channel and the blue light is something that's used very often.
You know what's crazy? I learned, like, doing all this and trying to develop everything, we learned that, and I did not know this, but that neurons are incredibly sensitive to light. Like, I mean, they just die from low levels of light. You have to give them nice media. You have to really take care of them. But they just do not like ambient light. It's fascinating. I never knew that before. I thought it would be like, oh, it's just a light. I'm not going to do anything. It's not like it's UV light or anything. But yeah, they're very sensitive.
That's why we got the brains on the inside.
Yeah, that's why.
Oops, how come those humans keep dying? Well, brains on the outside can't handle the light. That's just, What else are you working on in your lab? I'd love to know more, like beyond the ALS model.
Yeah, so we're working with some people to sort of recreate this process with things like tau and synuclein. So to go into Parkinson's and dementias, because we think that you can kind of take the same approach. It's, biology is pretty simple. If you can sort of reverse engineer these clumps that you form by trying to figure out what they naturally like to interact with, then I think we're in a good position to do that, or at least start questioning why these things form in the first place and try to figure out what pathways are going on. The other thing we're actually working on is what was mentioned earlier, is the nuclear transport pathway. So looking at how things are going in and out of the nucleus, because what we see is that in a lot of these diseases, especially ALS and FTD, it's really slow. So things going into the nucleus is just really slow compared to healthy stem cell neurons from a non-ALS patient. We don't know why exactly, but it looks like the membrane that makes up the nucleus is just not designed the same for some reason. Could be environmental, could be a bunch of other things, but it just doesn't seem to have the same rate of trafficking in and out as a healthy cell. It's almost like if you think of a tunnel, on the one side, there's a traffic jam. On the other side, things are fine. And so we're trying to figure out why that traffic jam is happening.
That's great. Is there anything else that you want to tell us about things that you're working on, or things that other things that you're interested in before we finish up for the night?
Oh gosh, I mean everything. So no, yeah, no, I mean I think the only thing I will point out is that just because I always feel bad whenever I talk to anyone about science in the lab is because like I didn't do any of the physical manual labor. And so I always want to point out that the people in the lab who actually did the work were the people on the paper. And then in this particular one, it was a student named Jake Mann. And there's a postdoc named Amanda Glexner and a technician named Joss Manua. And then we had good collaborators at like University of Pennsylvania who did some work for us from the shorter lab. So I just like to mention that because I always feel like they never get a chance to do interviews or say the cool stuff and get excited about it. They have to do all the grunt work and then they don't get to reap the rewards.
They'll get the love eventually. You're bringing it. This is No, it is important to be at the, you run the lab, but you rely on the people that you work with.
Absolutely.
You could not do what you do without the people working with the flies, working with the cells, making the optogenetics work. That's they're the ones that happens.
Yeah, and they're just so dedicated. They really care about it. It's not just for them. I mean, they don't get paid enough for it to be just for them. I know that for a fact. So they're just really interested in the science, and they're just a phenomenal group of people. So I just really have to tout that out, as much as I can. Awesome. And the other thing I will say, just from another standpoint, is that a lot of people who are interested or who are experiencing or things that are discovered in any of these neurodegenerative diseases are usually going to be applicable in some way to another disease. So anything that's found in ALS, I guarantee you there's some overlap with Alzheimer's. I mean, we as humans like to put things in these nice little boxes because it makes sense to us, but everything's kind of on a spectrum. And in my mind, you kind of have ALS on one side, maybe Parkinson's on the other, and Alzheimer's, FTD, and dementia somewhere in the middle. So, I just think that, anything we find, if we, and the other, sorry, I'm like keep going on here.
No, it's good. This is really fast. This is what you just said is super fascinating. Yeah.
Yeah. The last thing I'll point out is that, like, I think that the coolest and most interesting discoveries that have come out of the past, 20 years or so, have all been things that were kind of found in other simpler organisms or other simpler systems. Even the optogenetics, right? That was something that was found in algae. And then someone was like, oh, we should apply this to neurons. And that's now how we use optogenetics. So I think that, CRISPR was bacteria, right?
Bacteria, yeah.
Yeah. So I don't know. For anyone listening who's interested in science, if you have a cool idea, you should just really just try to apply it to something else. I think you probably don't know what you have.
Yeah. No, I think that's absolutely important. There's that curiosity to be able to start putting different paradigms together. And then there's also, I think you're absolutely right that we have this. I mean, I come from biology. I come from I must characterize what mammal this is. What bird is this? What is the species, subspecies? You got to go in its little boxes. And the same is to be said for the brain and its diseases and its disorders and the body in general. We have, these diseases that we specify by a name, but that could very often, like you say, be within a spectrum and related to others. And so I think that's a good thinking point to leave people on for the night.
Yeah, and also be extroverted, people. Because when you go out, because be extroverted. Go out and have beers with colleagues who are working on other things and have conversations and put these loose ends together that allow discovery. I mean, this doesn't, it sounds like this, a lot of this discovery doesn't come about unless there's not just collaboration, but crosstalk across disciplines. I'm sure your friend who was developing this probably wasn't even developing this to treat a disease, but this, this is how science is large collaborations. And I love everything about the story that you told today. It's
Oh,
a
thank
great
you.
example
Thank
of
you.
all of it. Yeah,
Hopefully it made some sense. I'm, my first
Oh, yeah.
absolutely.
live internet situation.
That was
Oh,
definitely that was the best explanation of prions I've heard. Where I remember in my undergrad and even since, every time I'm like, but what is it really? I feel like I actually got it this time, which is
progress. Yes.
No, very enlightening conversation. Thank you so much for it.
Yeah, thank you so much for having me. I really appreciate it. This is fun.
And if people would like to find out more about your research and more about ALS and other research that goes on in your institution, what websites should they go to?
So they could check out the, I'll probably just give you mine because I don't know all the actual addresses, but if you just do Donnelly Lab, one word, at donnellylab.com, I believe. That's a good question.
I will link to it on our show notes.
Yeah. Yes. But yeah, absolutely. That would be great.
You're like, what's my website? What's my name? Hello.
I have no idea.
Dr. Donnelly, thank you so much. Thank you for joining us tonight. Yeah, been absolutely great. Really enjoyed speaking with you. And I think I learned a lot. Blair learned a lot.
Yeah.
We all learned a lot. And I hope our audience did as well. And with show notes, we will have links to Chris Donnelly's lab website and also to the his other research affiliations, LiveLikeLou and others. So without any further ado, it is time for us to take a break. We'll be back after a few minutes of other stuff. We'll be back in just a few moments. More science coming at you. I've got more brains and yes, more brain things. All right, everyone. Thank you so much. We will be back with more This Week in Science. Thank you, Dr. Donnelly.
Can't explain the things you've heard with more than intuition. A line breeze that shows the way to go. New conclusion. The methods of hypothesis and patience are the only things I need. Put on a pair of goggles and go looking for the things I couldn't see. The answers lie somewhere within this scatter plot of first assumptions were correct. Disprove the rest. It's hot.
Everyone, thank you for listening to another episode of This Week in Science. I truly appreciate that you have brought us into your lives for another week to learn about science. I hope you enjoyed the interview that we just had. There is more science yet to come. But now, everyone, get your tickets if you're in the Portland area on April 3rd. We will be at the Alberta Rose Theater and hope to see you there. We will be joined by the PDX Broadsides and Dr. Allison Coffin to talk about hearing and how that works and the neuroscience of hearing. That's going to be very fun. Get your tickets. Information can be found at TWIS.org. Also at TWIS.org are all the things that you need to really suit your needs TWIS relatedly. Yes, that is true. All the related things. TWIS.org. Go there if you would like to find t-shirts and other items. We have a Zazzle store. Click on that Zazzle store link at the top of the page. It will take you to our Zazzle store where we have t-shirts, tote bags, pillows, mugs, hats, phone covers, all the things. We've got TWIS logo items, and we also have items that are artwork from Blair's Animal Corner calendars from several years now, which is very exciting. There's a lot of great artwork. I love the Kiwi tote bag. It's fantastic. Your purchases, a portion of them, do go to support This Week in Science. That's right. That's right. We get a portion of those proceeds. You shop and you support us. It's pretty awesome. It makes it really fun and easy for everyone. And then back at TWIS.org, click on that donate button on the sidebar. There's a yellow donate button that will take you to a PayPal interface. Whoops, didn't mean to do that. I'm going to shut that window down. There we go. We're going to the TWIS the PayPal interface that allows you to donate one time an amount of your choosing through PayPal. If you want to donate on a recurring basis, you can click on our Patreon link. That's at the top of the page. Click on the Patreon link. It'll take you to patreon.com/thisweekinscience. Click on Become a Patron and choose the amount that you would like to be charged once per month. That's right, one time per month. And really, the $10 a month category is one of the best because seriously, it's like a cup of coffee a week. That's about the same price. And you get a bunch of neat stuff. And we will thank you on air by name every episode that you are supporting us at the $10 category per month and above. But if you can't do that, think about, $1 or $2. Every amount helps. I mean, that's really it, is that your support helps keep this show listener supported and not advertising supported. Additionally, the more subscribers we have, the better we will do. All of us, the show, you, everything. Click that subscribe button if you have not subscribed yet. Or if you know someone who needs to subscribe to the show, tell them to go to twis.org and click the subscribe button to be able to choose between YouTube, iTunes, and Google for how they would like to subscribe. All 3 is cool too. We don't mind that. That's nice. Everyone, you help us do what we do. We could not do what we do without you. Thank you for your support.
Can't explain things you've heard more than intuition. A lot of reason shows the way to go. New conclusion. The methods of hypothesis and patience are the only things I need.
And we're back with more This Week in Science.
Yes, we are. We are back again to talk science. But first, it is that time in the show that we talk about this week in what has science done for me lately. Well, according to Beth Gilmore, she says, okay, I've got one. It may be slightly embarrassing, but it's for science that I tell my tale. This is kind of a life hack that I made up. Life hacks. They're good. Ready? Run out of deodorant? Caught out on a busy day? You've been running hither and yon and notice you stink.
It happens to me more often than you might think.
No problem. Hand sanitizer. It's small and you can carry it in your bag. Also, it's everywhere, on desks, in public bathrooms, all over the place. What science has done for me is taught me that underarm stink comes from bacteria eating and pooping. The hand sanitizer kills the things making the smell under your arms almost instantly.
Wow.
That beats any deodorant I've ever heard of or used.
Why does it feel to me like that would sting?
Because you're applying it to the wrong area, Blair.
If you're shaving, you might have nicks and cuts that would sting. But if you put hand sanitizer on your hands all the time, it doesn't sting
You know
your
what?
hands.
I can even cut to the chase there a little bit. I got ethanol where I work. The main ingredient of hand sanitizers tends to be alcohol anyway. Just do a couple spritz of those. Good to go.
I got some whiskey.
I got vodka. There we go.
Look, we are hacking next level right now.
Who sent that in, Kiki? I don't think you said.
Beth Gilmore, thank you so much for your life hack. And this would have come in very handy for that week when I ran out of deodorant and bought the wrong deodorant and it made me smell funny and I didn't like the way I smelled. I could have just used the hand sanitizer in my kitchen.
So that's an interesting thing that you just said, that it made you smell worse.
I had a deodorant.
I had a deodorant that somehow chemically combined with my natural microbiota, produced the scent of cat urine. like I put the thing on, I'm like, oh my God, now it smells like a cat peed in my armpits. This is so much worse than when I started with my natural man musk. Like this, you do need to be cautious with these
You
things.
can go the wrong
You
way.
can go totally wrong. Yeah.
I mean, I was having a very related conversation today at work, believe it or not, that I just kept saying, humans are supposed to smell.
Yeah.
I've certainly sat next to people on the bus where I'm like, no, you got to get a handle on that. But still, humans in general, we're supposed to have a smell.
Yes, we
We're
are.
supposed to be completely hermetically sealed.
No. All right. And on that note, thank you, Beth, for this wonderful life hack so that you do not have to keep yourself hermetically sealed, even though you might have run out of deodorant.
A good alternative to smelling like cat urine.
Yeah. Do you have other life hacks science has allowed you to discover and make your life better? Let us know. What has science done for you lately? Send me an email at kirsten@thisweekinscience.com or send me a message on our Facebook page.
Mhm.
And now, kind of apropos to your comment about us being hermetically sealed, no. Humans should not be hermetically sealed. According to new research, in fact, we should fly like pigeons.
Huh?
No,
What?
No.
We should have wings?
no, we shouldn't have wings, but a group of researchers recently published a study in which this is from Peking University in Beijing. They took 26 participants. So this is not a huge study. This is just preliminary, just kind of starting to get. And in one of the quotes of the researchers, they realize that their work is very early stages. But they've published it in an open access journal, eNeuro, so anybody with the link can access it. We will put it in our show notes on twis.org. What they have done, they took 26 participants, got them to sit in a dark room with their eyes closed. There's no sounds around them. However, the room is lined with electrical coils that could be turned on to induce a magnetic current. They recorded EEG activity, so electroencephalogram.
Encephalofa ding dong?
That's right. Electrical activity from the mass activation of the brain that ends up being recorded off of the surface of the scalp. So it's, a far cry from actually recording neurons in the brain, but it gives you an idea of activation in different areas of the brain. They recorded EEG activity in the brain while the magnetic field rotated around the room in various directions. Clockwise, counterclockwise, up, down, all over the place. What do you think they found?
I feel like their inner ear is such that it would work.
I think
Why
that
would
they
the
found.
inner
I think
ear
that
be
they,
such that it would work? Why do you say that?
Because don't we have little statolith hairs in there?
We have their calcium in the ears. The inner ears is calcium. Yeah.
I would have thought that the human skull resonated like an antenna, like a repeater for the magnetic frequency.
Yeah, so it's not that the skull itself reverberated in any way, but what they did find was that there was a definite difference between people exposed to changes in magnetic field versus people who sat in the same room with no changes in magnetic field. The EEGs responded to changes in the magnetic field. Now, these individuals came from Peking, which is in the northern hemisphere. So their brains responded to counterclockwise, swiveling in northern directions. But when the magnetic fields changed to a more southern hemisphere orientation, the brain did not respond at all.
What?
Yeah. And so the question is there a normalization of magnetic field based on where you're from? So you're in the northern hemisphere and your brain goes, this is normal. I will respond to this. They don't know yet still. Like they said, this is early stages. Also, there was a difference in counterclockwise rotation of the magnetic field versus clockwise rotation of the magnetic field. Why would the brains respond differently to clockwise versus counterclockwise rotation? That in itself is odd.
Yeah.
So,
I've never been to the southern hemisphere because there's like, I think only 3 places to go.
yes, only 3. Whatever.
Very
I haven't really done the math, but there's like, I assume like most humans live in the northern hemisphere. But maybe that's not. Maybe I'm missing.
Yeah, I think that's probably accurate. Yeah, majority of humans.
Yeah, that's a really odd finding.
So many things. So one, there's this background magnetic field that exists, but there are other things like living things that make their own magnetic fields.
Yeah, and there are magnetic fields all around us all the time because of our electrical wires.
Yeah, that's the first thing I thought of is right there was that, like, aren't we all always surrounded by and another low-lying magnetic field from our electronic
is like
house wiring? And
it seems confounding. It seems like there's so much that to be attuned to it at all would be more harm than good.
But the question is this, right? If you're attuned to it, oh God, sensory overload.
Too many things.
Great point. Yes. But at the same time, it's an interesting question. Do we have even the mechanism with which to perceive these magnetic fields? Like we've been talking about birds, sea turtles, other organisms that migrate and use the magnetic field to do their migration. And we've been talking about the fact that they have these over the years we've started realizing the mechanisms that are in place to allow it to happen. What are those mechanisms?
Right.
Are they the same in people? Is this really true? This study of 26 people? What are we looking at here?
Well, that's what I was thinking about. If I could just explain my inner ear hair theory a little bit further. I mean, they're all wavelengths, right? Sound wavelengths, magnetic and electric wavelengths. They're all wavelengths. I'm just wondering if it might be related. It seems like an odd coincidence if this turns out to be true and we have these structures in our inner ear, especially considering what we hypothesized to this point about pigeons, that there are similar structures. They're not made out of the same thing. They're not located in the same place, but we hypothesize that there are similar structures that help them observe magnetic fields. It's interesting.
It is. It's interesting. It's one of these things where you go, it's things that make you go, and can we study this a little bit more? I mean, as a physiologist, the first thing I think of, this is a sensory system. What is the sensory organ? What is the neuron that is in the eye, like you said, either the inner ear or in the skin or in the eye, somewhere that can be affected by the magnetic field? And what is its pathway and what does it affect? Or did it used to affect something previous to humans and it no longer serves a purpose in us?
I would oh gosh.
I mean, we could still have they are hypothesizing that there are sensory cells containing magnetite in neurons somewhere in our brains. And those sensory cells are what are doing this. So we could, but maybe they're leftovers.
Or maybe they just
when we were fish, who knows?
Maybe maybe they just didn't serve a purpose, but now can interact with electronics. So this is very quick, totally not to do with the story. When I was about 18 years old, I think, I heard a very faint, high-pitched sound. And I was trying to figure out where it was coming from. It was almost like a what is it, tinnitus kind of a thing?
Tinnitus?
Yeah, it was like but it was really like present, I found it was sort of directional. And I went outside my apartment, and there was a guy with like what looks like some sort of weird like Ghostbuster detecting device in his hand. I go, "Is that you making that sound?" And he clicked the thing and it stopped. I'm like, "Okay, it's off." He goes, "You could hear that?" What he was doing was he was with the cable company and he was looking for signal leaks. And I don't know what the device was exactly, but it was looking for signal leak. And for whatever reason, 18-year-old Justin could hear it. And it wasn't producing an audio. So
it was producing an audible sound, and your ears were younger then. And we know that younger people can hear much higher pitched sounds than older people.
But it was really like he was kind of tripped out that I could hear his like cable leak device, handheld device, and it was like absolutely obvious and prevalent to me. But I don't think it was emitting a tone. I think it was emitting a like what I was saying, like a wavelength of some sort.
Well, then Justin's a superhero. It turns out he's a mutant.
That my ear was translating into a sound because it didn't know how to define it.
Yeah, I just want to know, how did the magnetic field sound, Justin?
Like old man Justin tendinitis, like a vague, faint ringing in your ear, but directional. That was the weird part, is I could tell that it was coming from that a ways.
Yeah. Moving on from magnetic fields in the human brain, who knows? Maybe we'll see more of that. We'll see. We'll see what happens with this research, but it's interesting and thought-provoking. Organoids. I've talked about brain organoids on the show before, but this week, new study out in Nature Neuroscience, researchers in the UK at the Medical Research Council's Laboratory of Molecular Biology in Cambridge, they have developed mini brain organoids. So they took stem cells and they coaxed them into turning into human neurons, and then other cells like blood vessels and microglia and the other cells that all go together to create little tiny brains. Except one of the problems with mini brains is they start to grow into these little masses, and they don't have the blood vessels to reach all the way into the center very well, and so they end up dying and they can't last very long. And so this Cambridge lab has developed a methodology that they call ALICO, ALCIO, for air liquid interface cerebral organoid. Basically what they take the organoid and they slice it up and they put it in a dish, and so it's like got
and saute it in some butter and onions, or
air on one side, nutrients on the other. It's very happy.
Yeah.
Yes. And so this ALICO, these organoids, they have been growing them. They've lived for over a year. And so this is a big deal when it comes to these organoids, because normally longevity is not something that you see in the organoids. And like we were talking in our interview just a little while ago, there's that question of, you're going to develop a model system to look at, but then you're looking at these baby neurons. Right? I mean, maybe you don't get 55-year-old neurons, but the ability to grow a system that is at least several years old could be really helpful in understanding the development of disease. And so they have developed this culture that now has grown over a full year. Mini brain organoids. They've got back in November, I reported on the mini brains that were showing similar activation to embryonic brain tissue. Well, these little brains, these half-millimeter thick slices attached to a membrane, they let them grow their neurons out, and they were reminiscent of nerve tracts. And they were like, "Oh, yeah, well, just here's some mouse spinal cord. Do you wanna connect to that too?" And the little mini brain said, "Yes, yes, I do. I will connect to that." And so they grew their neurons and they connected to the spinal cord, and the spinal cord, of course, was connected to, skeletal muscle tissue of the mouse. And so these mini brains, without any prodding whatsoever, decided that they were gonna start contracting that muscle.
Mm.
Oh my God.
Whoa.
Yes. After 2 to 3 weeks in culture, dense axon tracks from the alicos could be seen innervating the mouse spinal cord, and synapses were visible between human projecting neurons and neurons of the mouse spinal cord. Evoked muscle contractions were intensity dependent, such that larger stimulation currents increased the amplitude of the muscle contractions. The mini brains were controlling the muscles connected to the spinal cords. That's
It's alive.
Yeah, pretty much.
So now I'm just starting to connect these dots here. If this would potentially be a good system to do some sort of ALS testing on, because it's better than just a dish of stuff.
Absolutely.
However, as you mentioned, it happens in older bodies. So mice die a little too quickly. They only live like a year or two, some of them. But he was saying that also horses get it, right?
Yeah, horses are
And
longer lived.
they live 15 to 20 years, a lot of them.
Yeah.
So we need some of these mini brains attached to horses.
So actually, but I don't know. My takeaway from that is kind of irrelevant whether the neuron is old or young. This is an effect that happens well beyond the even the health of the neuron itself. But what's really fascinating about this is that these mini brains are doing what we have not been able to recreate properly in people with spinal injuries.
Yes. Yep.
And that's
Mhm.
that's intriguing, that the mini brain will take the initiative that the human brain seems not to appreciate.
It's not that it doesn't appreciate it it's actually in humans specifically, when there is spinal cord damage, there are factors at play that actually stop reconnection. And so you have to get past those factors to be able to, like, kind of get rid of those damage factors to allow regrowth to occur. And so what's happening in this spinal cord, I mean, yeah, you're taking a spinal cord and saying, "Hey, new neurons, go connect." And they don't know that there's damage. These new neurons from the brain are like, "We want to connect to something we can talk to. This is what we do. We communicate."
Mhm.
You know, the spinal these mini brain neurons don't know anything else to do, because that's what they wanna do, is reach out and connect. They haven't been told not to yet.
Mm.
So there's
So,
a
I mean,
difference there, but I think you're right in that
there's
there's a
it
lesson to be learned in
sounds
how
like
to.
there's a therapy
Yeah.
in the maybe possible caveated future of
Yeah.
by utilizing some pluripotent mini brain
Just
cells.
You know what?
Stick 'em
Oh,
in
just
there,
put a
let
mini
them do the
brain on
work.
it. There we
Yeah.
go.
Slap a brain patch on there.
What's that on your side? Oh, that's Bob. He's my mini brain.
Mini brain patch.
Bob
Is
and Karen,
this how we end up with Krang in real life?
probably.
Oh, man.
Oh my goodness. Justin, tell me about God.
Well, according to some people, God created everything, which then leaves the obvious question: who or what created God? Now, thanks to science, we have an answer. Humans. Humans created God and gods.
Hmm.
And not just any old humans, but humans living in large, complex societies. In a study that runs contrary to how most God-fearing atheists might have conceived of the evolution of gods, that being that some good comes from a belief in an all-powerful something or another in order for civilization to get kick-started, an international research team found that large-scale societies precede powerful deities. The eye in the sky coming much later along in the evolution of those societies. The results are published in the current issue of the journal Nature. For their statistical analyses, the researchers used the Global Historical Data Bank, the most comprehensive and constantly growing collection of historical and prehistorical data, containing approximately 300,000 records on rankings of social complexity, religion, and other characteristics of 500 societies in the past, spanning some 10,000 years of human history. Quotey voice of Peter Turchin, University of Connecticut. It has been a debate for centuries why humans, unlike other animals, cooperate in large groups of genetically unrelated individuals. Factors such as agriculture, warfare, religion have been proposed as the driving forces. And one of the theories of the big moralizing gods creation hypothesis leading to society is that people begin to cooperate fairly towards each other if they believe in gods. They will punish them if they don't. And this is the reason for society's creation. Quotey voice here of Harvey Whitehouse. To our surprise, our data strongly contradicts this hypothesis. In most every world religion for which we have data, moralizing gods tend to follow, not precede, increases in social complexity. What's also interesting, though, in this is that standardized rituals of religion tended, on average, to appear hundreds of years before the gods were created, who would then later be the focus of those rituals. So it was sort of as if the whole God thing came in as a filler to tie in society and ritual and pre-existing tradition, and not as a thing that was the focus and creation and inspiration for those ritual behaviors.
So this is directly related to a theory that I've heard before, which is that large societies were the first time when you could have specialization. And specialization was the first time that people had real, legitimate leisure time. And with leisure time comes lots of sitting around and talking and thinking, which comes existential questions, which comes religion. And so it follows that pretty well to say once society started to kind of get its footing and figure out how everyone could work at their own specialty to make everyone work less, that then you kind of get this opportunity to move, to think inward a lot more than outward, which I think is where a lot of this comes from.
Sort of interesting. I kind of reminded me of God as a policeman. So as
that's what I was going to go for.
You have these social agreements and laws, and now you have a complex society, and you can't actually get police on every corner. That's much harder than convincing everybody that there's an eye in the sky watching you. And it sort of is, in that sense, then a precursor for law. But what this also says, though, is that we were already benevolent towards one another long before we created religion, and that this sort of collective identity was more important to creating a society than a eye in the sky sort of form of law. Like that came later as a fill-in, perhaps. as maybe the earliest form of government, but was less so important as an inspiration to begin a society or to begin. It's like that.
It's not like, oh, we all believe the same thing. Let's all come together as one and create some grand civilization.
Right.
That wasn't the movement. It was people already together, working together, making things happen.
Yeah.
On Blair's side, probably also coming up with different professions and tasks and having more leisure time and thinking, and also maybe getting in trouble more often. And you have these people living together, and maybe there needs to be a way to keep the peace and put it all together and hold it together in a way that isn't just military.
Right. But it reminds me of Christopher Hitchens commenting on "Thou shalt not kill," the Sermon on the Mount, when they finally announced for the first time, "Thou shalt not," and like everybody looking around going, "Oh, we should stop just frivolously killing one another." Never occurred to us that was a negative for our society. So Donnelly, like, this research obviously is illustrating that people got along great and formed very large, complex societies across the globe. I mean, this is an international study. They've got, if you go read the Nature journal article, you'll see this is spanning continents. That human society got along really well and was formed really, really nicely before the rule of religious law. And I'm also really fascinated because we already know this, that a lot of rituals that are considered religious had a pre-existing ritual, but that existed long before the religion itself was tagged as coming along.
The Christmas tree.
Yeah, like right. That's a great example of it. And there were even deities that had been associated with that also would show up later. But humans, whether you're religious or atheistic, I guess it just says we have greater bonds.
Yeah. It's a good place to start.
What's interesting from this, though, that they've got this huge database that they're looking at. Though it's not just looking at this, that now it's like this huge big data effort to study this,
so it's
where they're using where now we can look at different variables and see how they play out.
The CSHAT, I think, is what it's called, the database that archaeologists, anthropologists, religious scholars, people have been pouring data into this, and they're starting to look at this as being able to also map out other large scale evolutions in human history, and maybe look for connections that we didn't see before.
I think that's fascinating because I mean it's like I look at genetics and I go, yes, data, and then I think people and I go, ooh, messy.
Messy data on the outside.
What?
And I'm sure it helps that a lot of written history is somehow related to religion, so it's probably easier to track this kind of thing considering that as well.
Yeah, it's very exciting. Speaking of tracking things, what time is it? I think it's time for
finally
Blair's Animal Corner
with Blair.
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels.
What you got, Blair?
Uh-oh, don't look up. It's bird poop.
Ah!
So we've talked a lot on the show about coral reefs, how they are so important. Something we have not talked about on the show is, very much at least, is bird poop. And University of Otago in New Zealand looked at the impact of bird poop on coral reefs. You guessed it. What they found was that seabird nutrients through poop are assimilated by corals and enhance coral growth. So an important part of the story in the coral reef is indeed bird poop.
So
Really?
you also brought a story a while back about shark poop being crucial to the corals.
So let's take a step back, everyone, and just remind ourselves that no digestive system is perfect, which means whatever an animal is eating, there is a lot of good stuff left in their poop, which is why there are so many animals that are coprophagous that eat their own poop, is because poop has a lot in it. It's something that we shy away from evolutionarily because a lot of the time it has bacteria in it. That's why we think it smells bad. But those bacteria signal us
they're eating poop.
They're eating poop. There's a lot of animals on this planet that eat poop. All detritivores eat dead stuff and poop. Decomposers eat poop. So poop is actually something that is a really important part of nutrient cycles on our planet. And a lot of the time we look at it and we just think of it as one thing, as waste. But it's actually a really big part of the story of an ecosystem, is where that poop is going, because there's nutrients being stored there. Whether they be seeds that are going to become future trees in some cases on land, whether it is hippo poop that's bringing nutrients into the Nile River, or whether it's the shark poop saving ecosystems underwater. In this case, seabirds, their poop has been proved to be an important part of the coral reef equation. So this research was conducted with 2 Fiji marine protected areas, one remote island with an intact coastal forest with breeding seabirds on there, and the other away from seabirds and their associated guano. Natural chemical tracers in coral tissues showed that corals growing near the roosting seabirds had seabird nutrients, as you might expect. A 1-year growth experiment showed that the corals grew up to 4 times faster when poop was nearby compared to when it was not.
So for those not familiar with multiplication, 4 times is approximately the same as 400%.
400%, yeah. So this is interesting for a few reasons. One being, a lot of people don't realize coral reefs, and this is going to sound obvious once I say it, the water there is pretty poor. When you think about a coral reef, it looks beautiful and pristine and crystal clear. When you think about a body of water like one of my favorites, the San Francisco Bay, you think about green muck. It turns out the San Francisco Bay is actually way more nutrient rich than those coral reef ecosystems, because that muck is actually nutrients. So when you think about coral reefs, the reason corals have zooxanthellae in them, this photosynthetic algae, is because there's so few nutrients that even though coral is a little animal that can filter feed through the coral, there's not much out there for them to grab. So the algae is actually, the zooxanthellae is subsidizing the lack of food that they would need. So when you have a bird eating far away and then coming and pooping near the reef, that's adding nutrients back into an otherwise nutrient poor water table. So this makes perfect sense to me. But it's interesting for a bunch of reasons. One being that when we are considering saving coral reefs, seabird populations are not part of the equation. Why would they be? Turns out, once again, this is a situation where saving a species or an animal is not enough. You have to save the ecosystem. So a protected area that doesn't scare away seabirds is an important part of saving coral reefs. If you have ecotourism going on with coral safe sunscreens and no motorboats and all this other kind of stuff, but you still have humans snorkeling nearby that are scaring away the seabirds, that can still cause problems to the coral if you're scaring away those sources of nutrients. So that's one side, is protecting the wild coral. The other side of this is that there might be part of the equation when we're raising coral in captivity that we haven't considered. So it might be time to go start harvesting bird poop.
Well, I just wonder also, I mean, is there a difference in the quality of the guano between different species of birds? I mean, gulls will eat anything. They are like the pigeons of the sea. And so are the gulls going to be differently producing for the coral versus skuas versus petrels versus, seabirds?
That's a
Which
great
seabirds
question.
are better?
Yeah, animals that feed primarily on bottom feeders, are their nutrients available in their poop as good as birds that feed on swimming fish?
Yeah.
Maybe theirs are better. We don't know. Or better than those that feed on kelp. I don't
But what I think the big story is that, like what you're saying, is that overall the water is nutrient poor. And so the poop is adding nutrients to the water. So if we can figure out what nutrients to add to the water, we don't need to go harvesting guano necessarily.
Right.
We don't have to have big seabird farms
Yeah.
for guano farming for our coral production needs. But what could we add to water that we aren't?
Yeah, absolutely. And in this case, it's not saying necessarily that seabirds know seabirds makes or breaks a species, but it does impact the growth rate. So especially when we're thinking about coral reefs that are trying to rebuild after some issues, if it's a warming instance like in the blob, or if it's in some sort of acidification event or an oil spill, especially if it's a coral that's trying to rebuild, or if we are trying to build corals artificially away from the reefs in the wild and transplant, if we can grow them faster and healthier, that is really important. But I think that the bigger takeaway, in my opinion, is reminding everybody that the entire ecosystem is the part that needs to be saved. And so even when we think we might be doing what's best for an ecosystem, if we are displacing any member of the food web, there are potential impacts.
Amen. There we go. Tell me another story.
Oh, I just want to tell you a story of the world's oldest sperm.
Wait, what?
Deep in a laboratory in Sydney, Australia, there is a vial of 50-year-old sperm stored in liquid nitrogen.
Okay, so is this sperm from a 50-year-old? Or is this sperm that has been stored for 50 years?
This sperm was collected in 1968.
Wow.
It was
sperm.
It was collected from a very important sheep. This is not human sperm. I'm trying to find his name in the study. The sheep had an amazing name. I'll find it in a minute. But this amazing sheep has done something really amazing. Post-mortem, this sheep has impregnated 34 ewes. And the resulting birth rate was the same as sperm that has been frozen for only a year. What's more, the lambs born from this 50-year-old sperm appears to display a body wrinkle that was common in sheep of the 1960s.
Interesting.
It was originally selectively bred to maximize skin surface area so that it would increase wool yields, but it fell out of favor because it was difficult to shear, and they often accidentally nicked the sheep in the shearing process, which a lot of people don't realize. A lot of wool farmers are very protective of their flock, take care of their sheep very, very, very well. A happy sheep grows more wool. So that is something that definitely had to fall out of favor. So this, not only was the sperm viable, but they confirmed, yes, you can see here what sheep looked like 50 years ago.
That's awesome.
Yeah, so this has a bunch of implications. The one that jumped to mind for me first was that the sperm vault, much like the seed vault that we always talk about, is something that could happen. This is something we have started thinking about, and people have started collecting semen samples from animals that are endangered, that there is a fear that they will become extinct someday.
And someday when men are extinct, we will have their sperm in a vault.
Yep, sure. Absolutely. But in the meantime, perhaps we could bring back the Sumatran rhino someday. Who knows?
Maybe. Yes.
But this is something that is really surprising, that 50 years of storage in liquid nitrogen, and this thing worked. So they had to thaw it after storing it at negative 196 degrees. They took in vitro tests on the sperm quality to determine motility, velocity, viability, and DNA integrity. All of it was good. There was no difference in any of those tests between the sperm frozen for 50 years and the sperm frozen for over a year. Out of the 56 ewes they inseminated, 34 were successfully impregnated. They did a recently frozen sample, just 12 months old. They inseminated 1,048 ewes, of which 618 were successfully impregnated. So the pregnancy rate was 61% for the old sperm, 59% for the recent sperm. There you go.
Old sperm is better.
I mean, it's the same. It's statistically the same.
A little bit better.
His name was Sir Freddie. He was born in 1963. He was one of 4. There were 4 rams that donated to this cause. So this is really interesting because it has the integrity of frozen sperm for future efforts in conservation. But the other thing that I thought was really interesting is that these sheep herders were particularly interested to be able to see differences between old lineages and current lineages. And so this might actually be useful in figuring out how genetics has changed over a 50-year period or longer. So you can see how things have diverged and adjusted over time by using a very old frozen sample.
Yeah, that's great. I love it. I mean, it's the idea of potential, lots of ideas. Yeah, genetic analysis, reanimation of extinct or species that are going extinct, being able to save them, like the seed vault. But then it also kind of gets at that idea of, we talk about wanting to potentially bring back species like the mammoth, and they've been frozen under ice in Siberia and other places. And it gets at those mechanisms of what's happening or not happening while things are frozen.
And then we could finally find out how important its poo is to the
Yeah, absolutely.
That's right. Any more stories, Blair?
Oh, I'm all done.
You're all done. All right. Asteroids, you guys. Quick news for the end of the show. Very quickly, just want to remind everyone. Science and Nature published a bunch of studies this week related to the Japanese Hayabusa 2 mission to Ryugu and also the mission to asteroid Bennu. There are different analyses that have been going on to look at these different asteroids to get different pictures of solar system formation, how the asteroids have been evolved. The Ryugu, interestingly, is not much denser than water on its own, although not much water has been detected. It's really not dense. It's a very interesting, not dense asteroid. Bennu is a bit more dense. It's kind of it's got a similar density just to gravel as we would find gravel here on Earth. Interesting aspect of Bennu, though, is that it seems to be irregularly just spitting off rocks and dust and bits of its surface, and nobody seems to know why or how. And so there are some questions now about these very interesting asteroids. Japan's Hayabusa 2 mission will be a return mission. Hopefully, we'll get some interesting insight into what's going on in Ryugu. Researchers at MIT are busily trying to create gray goo. Haha, awesome. So gray goo is it's science fiction-y compound, robotic compound of these parallel processing robots that work together to basically take in any resources from an environment, break them down, and turn them into anything else.
The
They are
blob. It's basically the blob.
Yeah, the blob. It's little machines, micro machines that can become anything. Yeah, you need a new arm? Let's just make a new arm. I can do that. I'm going to take some of this dirt and this other stuff, and I'm going to make it into an arm.
Oh, it's a T2. That's what it is.
Yes.
Oh.
Yeah, so the gray goo researchers it's not the gray goo, but researchers at MIT and Columbia University School of Engineering and Applied Sciences are equating what they're doing with what they call particle robots as almost being gray gooish, in that they have modeled up to 100,000 of these little robots working together to move and to move other objects and to do various tasks, but not that any one of the robots would actually know what was going on. Each of the little particles, in the same way that cells in an organism would work together to create the activity of the whole. So these little particle robots, they're saying, is the wave of the future in terms of modular, adaptable, useful robots.
Terrifying.
Super fun. Well, the video, they're really in the video from Columbia University School of Engineering. They're these really cute little green robots. I think they're really cute.
They look like a
flowers.
slime mold.
No, they are kind of small. And so that's another thing, is maybe what these researchers are doing is not really creating gray goo, but recreating slime mold.
Yes.
Yes. Justin, you got one last story?
I do. This is the story about hepatitis B doing some science. Australian researchers have used that virus to figure out how humans got to Australia and about when they did. So I guess the hepatitis B is sort of an endemic problem amongst Aboriginal peoples of Australia. And something called the Characterizing Hepatitis B in Indigenous Australians Through Molecular Epidemiology study, aka CHARM. They made a pretty interesting discovery, which is that the hepatitis B that's found there is unlike hepatitis B anywhere else in the world. And in fact, there's sort of two unique strains of it that are broken up amongst two large populations of Aborigines who also happen to speak different dialects of Aboriginal language. So it's very, very clear that this came with and split with these peoples a long time ago. Utilizing the same sort of thing that they do when they try to backtrack a human genome to figure out how old it is, they use this virus to backtrack how old the virus got to and split from itself. And their findings show that the Aboriginal Australians arrived in Australia about 51,000 years ago. And it would also indicate that it took about 8,000 years to get to Australia. So pretty interesting use of a virus to track human population transfer. And just a little side note, as hard as this study is trying, I still feel like the Australians are racist. So in the actual, this is the press release. This is from the University of Melbourne. It says this adds weight to the theory that the mainland Aboriginal population separated from other early humans at least 59,000 years ago. What do you mean other early humans? Wouldn't it just be from early humans?
Early humans.
Not other early humans? They're not. And they also refer to the this particular hepatitis virus as all right. The researchers call this a precursor of the modern virus. Like, no, these are current humans. Stop it. Stop layering your racist view of what an indigenous people is. They're not an other early human. They're current humans. You're not a modern human. You're just a current human too. Okay? There's no such thing as a modern human. There's just current humans. That's the only kind of human that can possibly exist now is the current ones.
Which is
fair
true.
Point. Fair point.
Yeah, no, it's true. But the Australians continue to be racist. Even in a study where they have the best intention of curing hepatitis B amongst Aboriginal people, and the study was even had participation from like 40 different Aboriginal groups. They still managed to be racist, like blatantly racist in their copy, in their press release. Other early humans. Stop it.
And the wonderful famous words. Yeah. The famous word, stop it.
Stop it.
Stop it. That's one of my favorite SNL skits from way back. Bob Newhart. He's a therapist. Woman comes in and says, I've got all these problems. He goes, stop it. Well, I keep doing this. Stop it. It's the best. Yes, just stop.
Or the more modern, really?
Really? Yeah, exactly.
Another good
one. The brilliance. All right. Speaking of other shows, this is this show. We've come to the end. Thank you, everyone, for joining us for another episode of This Week in Science. We hope we brought you some interesting tidbits of science news, some thoughts that are going to keep you pondering for the rest of the week, some things that'll make you curious and make you delve for more information. We hope that you come back again next week. I want to say thank you to Fada for helping out with show notes, for helping out with social media. Thank you to Identity4 for recording the show. Gord McLeod, Ben Rothig, thank you for chat room help. Thank you also to our Patreon sponsors. Thank you to Paul Disney, Richard Onimus, Ed Dyer, Stu Pollock, Philip Shane, Ken Hayes, Harrison Prather, Charlene Henry, Joshua Fury, Steve DeBell, Alex Wilson, Tony Steele, Craig Landon, Mark Bizarros, Jack, Matthew Litwin, Jason Roberts, Bill Kay, Bob Calder, Time Jumper 319, Eric Knapp, Richard Brian Condren, Dave Neighbor, Aiden Jeff Bont, John Bertram, Mountain Sloth, Seth O'Gradney, Stefan Alberon, John Ratnaswamy, Dave Friedel, Daryl Myshak, Andrew Swanson, Paul Ronovich, Corinne Benton, Sue Doster, Dave Wilkinson, Ben Bignell, Richard Porter, Noodles, Kevin Reardon, Christoph Zucknerek, Ashish Pants, Ulysses Adkins, Sarah Chavis, Artyom Rick Ramis, Paul John McKee, Jason Olds, Brian Carrington, Christopher Dreyer, Lisa Slazewski, Jim DePoe, Greg Riley, Sean Lamb, Ben Ruthig, Steve Leisman, Kurt Larson, Rudy Garcia, Marjorie Gary S., Robert, Greg Briggs, Brendan Minish, Christopher Rappin, Flying Out, Aaron Luthen, Matt Sutter, Mark Hessenflow, Kevin Parachan, Byron Lee, and EO. Thank you for all of your support on Patreon. For those of you who might be interested in supporting us on Patreon, you can head to TWIS.org and click on the Patreon link. You can find out more information there. Also, you can help us out by telling people to subscribe to TWIS. That would be great. If you're in Portland, don't forget that we have our live show at the Alberta Rose Theater, April 3rd find information at TWIS.org. Next week, we will be back broadcasting live online at 8 p.m. Pacific time at TWIS.org/live. You can watch and join our chat room. We're going to have an interview with astrophysicist Katie Mack. And she's going to join us to talk about black holes and her recent study. It's going to be fantastic. But if you can't make it, don't worry about it, because this is a podcast and you can listen to the recording on your device if you're subscribed, or you can find past episodes at TWIS.org or our YouTube channel.
Yeah, thank you for enjoying the show. If you're looking for that podcast that Kiki mentioned, just Google This Week in Science in your iTunes directory. Or if you have one of the mobile type devices that is not Apple affiliated, you can look for TWIS, TWIS, the number 4 Droid app in the Android marketplaces. Or again, anything Apple marketplaces, just look for This Week in Science.
For more information on anything you've heard here today, show notes will be available on our website. That's right, it's at www.twis.org, where you can also make comments, start conversations with the host and other listeners, and find out all the information about our live show. Come on down and we'll sign all of your TWIS merch.
Woohoo!
Yeah. But if you can't make it, if you don't want to just mingle with other people who are listening to the show, you can just talk to us directly. Email Kirsten at thisweekinscience.com. She always responds. Justin at twistminion@gmail.com. I sometimes respond. Or if you have any form of animal question, no matter how strange you think it is, you can email Blair at blairbaz@twis.org. She has the answer. This is my go-to with every strange question. Just be sure to put TWIS, TWIS, in the subject line of whoever you are emailing. Otherwise, your email will get spam filtered into oblivion. You can also hit us up on the Twitter, where we are at TWIS Science, at Dr. Kiki, at Jacksonfly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to you tonight, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
Yes, yes, yes. But if you have learned anything from the show, please remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. This week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. Cause it's This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. 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 I've got one disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma. Got the cause it's This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. 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 I've got a laundry list of items I want to address. From stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. So how can I ever see the changes I seek when I can only set up shop 1 hour a week? This week in science is coming your way. You better just listen to what we say, and if you've learned anything from the words that we said, then please just remember it's all in your head. Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. 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 This Week in 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 Science.
And that is the end of the show. Another show. And why didn't it stop? Why didn't it stop? It's the end of the show. The show. Clack, 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 clack. Isn't that a nice postcard? Look at that.
Woo!
Look at that.
Can you send that to me and I'll social media it?
Oh, yeah. I need help. We need to sell tickets. We need ticket sales. Yes, I will send that to you. Do I have it on this computer? See if I can send it to you right now instead of waiting. Yep, got it. Okay.
Great. I'll do it tomorrow.
Awesome. Hey, everybody. How are you doing out there? That was a good interview. I enjoyed
that was fun.
Yeah. Chris was sweet.
He did great.
Yeah.
He's good at communicating his science. It was
I
cool.
thought he was. Yeah. And even though he was nervous, it didn't really, it didn't come across in a negative way, which sometimes it can be not as great.
Yeah.
No, he did great. Identity 4 is going to take the train down. I love that.
Gosh.
I think that is
so
just
so many TWIS staples in one place.
I know. It's going to be fantastic. Yeah. I need people to buy tickets, though. We are not doing great on ticket sales yet, and we're 2 weeks out. Still 2 weeks out, which is fine. We need to sell. We need butts in seats.
So I will say I tried to find it through the Science Talk website.
Oh,
and
but haven't I sent you links?
Yeah, no, I just wanted to see. I was like at work and someone was asking me about it, and I was like, oh, let me see if I can just find it real quick.
Okay.
It was kind of hard to find through Science Talk.
Yeah.
Just
So maybe it's just multiple places, but not a direct.
Yeah.
Bye, Adam Weaver. Have a good night. I want to see Love, Death, and Robots.
Yeah, I just watched a few episodes tonight.
Really?
It was really good.
I haven't seen any of it yet.
Yeah.
I don't watch anything anymore because my child is awake, and then I go to and I put him to bed, and I just don't. I read after he goes to sleep.
I will tell you,
TV.
There's lots of swears and there's some light nudity in that show, so I would not approve it for Kai for sure.
What? Light nudity?
Love, Death, and Robots.
Love, Death, and Robots. It's really good. It's really good. It's just definitely adult.
So, Blair.
Yeah.
I have a mission for you.
Okay.
It's due by noon tomorrow.
Oh no.
Yeah. I need a
tight one.
This conversation that I prodded you with before is now escalating.
Fecal plugs.
Yes. So and it came to not not hibernation, but winter sleep.
Yes.
Wanting to know curiosity around the evolution of winter sleep and what animals engage in this activity.
Oh my God, so many.
Bears.
Oh, you mean winter sleep that is not hibernation.
Right. Like what is see, because my take was like the polar bear thing. Of there being a necessity of hiding the young from the other polar bears might have been the inception of winter sleep. It might have been the precursor reason for winter sleep before it became just a thing that all bears just do.
I would say it is more likely that North American and just kind of Arctic adjacent, not Arctic, but Arctic adjacent species that hibernated continued getting larger. And eventually they were too big to cool down enough to undergo full hibernation. That would be my hypothesis.
So what critters would you say? Just, I mean, again, you have till noon tomorrow to come up with the correct answer. But off the cuff, what other animals do this winter sleep behavior? Is it just is it just bears? And that smaller creatures hibernate? Or like, what else?
That's a really good question.
What else does this?
Yeah. So this is kind of back to that whole conversation of like humans wanting to categorize things so badly and everything is a spectrum. So it's your question of what the cutoff is. What is the cutoff of the temperature or metabolic rate change that constitutes winter sleep versus a full hibernation? Because my understanding is there's a lot of animals that hibernate or go through estivation or some sort of torpor, drying out and sleeping.
One of the examples was given was like a frozen toad that you could warm up in your hands and then it comes back.
Yeah, those alligators that were poking their noses through the ice, right? There's a huge amount of insects that when things get really dry, they just burrow underground and they hang out. There's hedgehogs are one of my favorite examples. They go through estivation, which is hibernating when it's warm out.
Interesting.
So there's the whole lizard conversation we had before. Lizards hibernate. So I will think on it, but nothing is coming to mind.
Okay. Interesting. So what other, but okay. So let's limit this quite a bit. So bears winter sleep. They're big. They're way too big to hibernate. So they go into this winter sleep regenerative mode. Is there anything else in the Arctic adjacent that is doing a similar activity?
Say it again. Ask your question again.
So is there another large animal that is doing winter sleep? I mean, caribou aren't. They're up running around. They don't care. They're not going to.
Yeah, and I think that's the problem. Is this whole idea of conservation of heat? Is that when you lower your body temperature too much and you're huge, it's really hard to get your body temperature back up.
So I mean, is it like Arctic hares running around all year round?
They're little though.
So is there what else does any form of anything close to what a bear does, and is an animal larger than a hare?
I mean, it's possible things like ground sloths did it. Giant ground sloths in the past. I don't know. That's a good question.
We don't really have another example outside of bears. I'm astounded by the uniqueness of their winter sleep ring. Then again, you have till noon tomorrow, at which point I expect a full report. But so this is a very uniquely bear activity, because when people say what hibernates, everybody thinks of a bear, and that's obviously not hibernation.
Yeah.
As we have been reprimanded by Blair. It's called Winter Sleep.
Mhm.
But is it so unique to them? Is there anything else?
Yeah, I don't know.
How? Blair is stumped. I have not seen this.
Maybe there just aren't any, Justin.
No, that's perfectly acceptable too. I mean, that's a fine answer. Though the idea that winter sleep could be a thing, and it's just not bear. It's just bear version of hibernation. How bears hibernate? I'm just trying
So
to
okay, so the research I'm finding just right now in a very short dive is that the thing that really truly distinguishes the winter sleep from hibernation is that during true hibernation, as I said, the animal's body temperature drops to match the outside temperature. Their heart rate and rate of breathing slows, and they will not wake up when they hear a loud noise or even if they are moved or touched. While in torpor or winter sleep, the animal can wake up quickly and easily.
But they don't need to say the animal. They just need to say the bear. Unless there's something else.
So hold on here. Let me just see here because I think Tenrex.
This is a creature I have no.
Go through. I don't know if they do torpor or if they do.
How do you spell torpor?
Hibernation. TORPOR. Or hibernation.
So currently in our conversation, torpor only applies to bears.
No, because I think torpor is a very wide net. So some of those insects, for example, are going through torpor and not hibernation, but it isn't due to cold and it's not due to winter.
Interesting. Okay. So, but that's such a big gap evolutionarily then.
Woodchucks.
Ooh.
Woodchucks are hibernators.
Yeah, hibernators. So I think this is part of the problem is that torpor and hibernation is used kind of interchangeably.
Even though it's not interchangeable.
Correct.
Yep.
I like how we all have the hand to mouth.
Hibernation in mammals. Good night, Fada.
I think this is tough. What is your
Oh wow.
Estivation?
Sarah Forfar is joining us from Zimbabwe. Hello, Sarah.
Oh my.
We are discussing animals that hibernate versus those that have winter sleep. And
so far, all we have in winter sleep category is bears and some insects, which might not be in the winter.
Well, yeah. So that's why it's not winter sleep. That's why this all gets confusing because there's also estivation, which is like hibernation, but during hot and dry periods.
Which is I would guess, without having a Google in front of me, is a thing that you would see in
true hibernators: woodchucks, ground squirrels, and bats.
Ground squirrels,
turtles, also. Yeah, this is going to take a
minute. I like this. Common poorwills are the only species of bird that hibernates.
Oh my God. Related questions. Can turtles pee out of their mouths? Thanks, Google.
Why is that a related question? Do they hibernate? Do they pee out of their mouths?
Which actually, I think we
No.
reported on that on TWIS. That was a Chinese turtle that we found can release urates out of their mouths. So,
Oh my. Dear mice, short lives, torpor. That's not hibernation. That's torpor.
See, this is what's hard is that people use torpor interchangeably.
Yeah.
So some places use it interchangeably and some do not.
Common porwills are the only bird that goes into true hibernation. I like knowing that.
Turns out all the frozen mammoths we found,
Pretty.
they thought they could hibernate, but they were wrong.
Right. I'm just going to go to sleep for a while. It'll be just fine.
Okay, so according to thoughtco.com, which I don't know how reputable the source this is.
Not.
Yeah, I know. Hibernation is a voluntary state excuse me, that an animal enters in order to conserve energy. My body's like freaking out. Think of it as a truly deep sleep. It's a body state marked by low body temperature, slow breathing, heart rate, low metabolic rate. It can last for several days, weeks, or months. And it is triggered by hormone changes.
So I've heard of the summer and dry thing in amphibians, that they can go into like they dig down to the mud and do like but outside of bears, this was I mean, we went through, I think, the same struggle here, except that I was like, as soon as I talk to Blair, she'll have the answer.
Yeah. So this is saying that unlike hibernation, torpor is involuntary as conditions dictate, and it lasts for short periods of time, sometimes just through the night or day, quote unquote, hibernation light.
So wait, hibernation is voluntary?
That's what this says, but
it's kind of,
I mean, so it is and it isn't. The bears at the San Francisco Zoo don't hibernate because it doesn't get cold enough.
Okay.
But then again, like you were talking about, they don't really hibernate anyway. They go into a torpor-like state, and it's related to temperature. On Wikipedia, it's saying that obligate hibernators are animals that spontaneously annually enter hibernation regardless of an ambient temperature and access to food.
Interesting.
So that's many species of squirrels, rodents, mouse lemurs, hedgehogs, insectivores, monotremes, marsupials. Yeah, and then their body temperature drops to near ambient temperature. The typical season is characterized by periods of torpor interrupted by periodic euthermic arousals. And nobody really understands why they kind of wake up and their body goes back to normal again and why it goes back down again. Nobody understands it.
So now I'm finding a bunch of articles that say that hibernation is an extended period of torpor.
Yeah, there's that idea also. Yeah.
So, Justin, here's the problem. There's not really an answer.
Because not a good answer to any question.
There are a bunch of animals that somehow go dormant for part of the winter. Okay. There's a huge amount. So the question is, for how long? How far does their body temperature drop? And so based on that, you can kind of move forward. But I think there's actually still, we're not sure. We know for sure that bears readjust, wake up, move around, eat, do other things if they want to in their winter sleep.
So if I'm getting the short list of answers that I'm looking for, right now all I have is bears and ground squirrels.
Well, ground squirrels.
Is that fair?
So the ground squirrels are tough because we found out that they go through torpor.
Mm-hmm.
We don't know how long of torpor, which means we're not sure.
Okay.
Eric in Alaska says that bears enter winter sleep more due to food availability than cold weather.
Yeah.
And they have bears that are seen out and about in the winter because there's human garbage around, and they have to keep that in mind.
Interesting.
because people are around, there's food around, garbage. And the bear's
No
like,
need to go wait for the spring bears.
why am I going to get all skinny?
Hmm.
Especially if I need to have babies. Yeah. That's interesting.
So, and I think this is part of the problem, Justin, is that like, unless you speak to somebody who studies metabolic states in mammals as like their main focus, you're going to have a lot of trouble figuring this out because people use the words so interchangeably. So I just found 20 articles that say that ground squirrels hibernate.
Yeah.
And then I found one article that said that they go through a light hibernation where they wake up a bunch, which would mean they're not hibernating. So who's to say?
I'm glad I asked the question. It seems like it must be a good question because you were trying very hard to massage your brain.
Yeah.
Yeah.
So this says BBC, which I think is a pretty reputable
They're
source.
British. They run the gamut.
Says that they
ridiculous
drop
to-
Arctic ground squirrels drop their body temperature below freezing. So I would say, in my understanding of the definition, that is hibernation.
Yeah. Yeah.
So not helpful to you.
Yeah. So it takes them out of the category of the winter
Yeah.
sleep thing that
Yeah.
bears do. But you could just then I guess that like what so we don't have a definition of what bears do outside of what bears do, just because of their size in relation to everything else. So bears hibernate. It's just that when you're that big, hibernation is called torpor.
So torpor. So winter sleep and hibernation are both kinds of torpor. Torpor is when you reduce your metabolic rate and your body temperature.
Which I need to do now.
Yeah.
So say goodnight, Blair.
Good night, Blair. Say good night, Justin.
Good night, Justin.
Good night.
Good night, Kiki.
Good night, everyone. I will see you next week. We have 2 weeks until our live show in Portland. So, Justin, did you get your plane ticket stuff? Did you get an email about that?
No.
Do you have the day off work?
Yeah, I have the days off work. I don't have any email or any kind of confirmation anything.
Check your email, man.
What email did you?
TWIS Minion. I sent it to your email, man. Check your mail. Yes. We'll figure it out before then, I'm sure. Everyone, have a wonderful week. I will do my Twitch thing on Friday, but otherwise we will be here next week. Can't wait to talk to Katie Mack next week.
Yes.
Maybe her co-author, Robert McNeese. We'll see how that goes. It'll be fun. We'll talk black holes. It'll be amazing. To infinity and beyond, everyone. I have to press buttons and turn this thing off, don't I?
I wonder if I blocked the airline.
You might have.
I might have. Yeah. He sent me a lot of junk email.
Junk mail. Okay. Not tomorrow, Ed, on Friday. On Friday. And it looks like I'll be on Daily Tech News Show next Wednesday.
Ooh, that's exciting.
Yeah, I reached out to Tom and he was like, come on the show. So this happened while this show was happening. It's amazing.
Exciting.
I know. All right, everyone. Have a good night. I'm clicking stop broadcast right now.